All right, hello, absent students. Welcome back to biology. I want you to go ahead and follow along this set of notes. You can stop it or speed it up as much as you want, but you should be completing the note sheet at the same time. Some of you might want to do a split screen where you have me and my video on the left side and the note sheet that you're completing on Cami on the other side. Let me know, email me if you have any questions or you're having any trouble doing this. All right, we are starting a new unit. If you haven't taken the evolution test, don't forget that you need to take the evolution test when you come back, when you return from quarantining. Okay, and if you're staying away because of COVID numbers, you still have to take the evolution test. So you'll need to contact me and we'll figure out how to get that to you, how to get you a test. All right, moving on. We are starting a new unit. It's called genetics. I'm super excited about genetics. I think everybody's kind of excited about genetics. So let's start with the notes quickly. What is genetics? Genetics is the study of how traits or characteristics are passed from parents to offspring. So you fill in the blanks as I'm going. All right, moving on. Oh, another handsome fellow. Remember, we had a handsome fellow Darwin in our unit on evolution. Well, now we've got this handsome fellow. His name is Gregory Mendel. Before anyone knew anything about DNA, Gregory Mendel began to study patterns of inheritance in plants. He was a monk and he lived in an Austrian monastery, which is a religious building where just men live. And he spent his time gardening and he would purposely cross different types of plants. I'll teach you how to cross plants in just a minute. And then he would look at the offspring and he made notes and he kept really detailed notes of what was happening in his gardens. And he was really considered the father of genetics because of the work that he did. So his discoveries became the foundation for the field of genetics. What is it that he did? Well, let me give you one example of something he did. He had hundreds and hundreds and hundreds of plants and experiments and things that he worked on. So this first one is just an example. He crossed purple and white flowered pea plants. Did you know that pea plants have flowers? Well, they do. And some of them have purple flowers and some of them have white flowers. Now, next to it, it says it's the P1 generation. That means the original parental generation. And it means that they're always purples or they're always whites. Don't worry about it right now. We'll come back to that idea a little bit later. So it says, the question says, what do you think the offspring look like? If you cross a purple flower with a white flower, what are their babies going to look like? Now, just to remind you, I know with plants it's kind of complicated. What's a baby plant? A baby plant is a seed and then you grow it and it turns into an adult plant. So what did the seeds, when they grew up, what did they look like? Did they have purple flowers? I'm giving you time to think about it. Did they have white flowers? Sometimes students say lavender flowers. Sometimes we think pink or purple and white flowers. What did it look like? We'll come back to that question. Before we can go much further, we need to talk about how he was crossing them. How is it that flowers mate? Well, I don't know if you remember this, but plants, the part of a plant that's sexual, I guess, is their flower. And they have eggs that are hidden inside the female part of the plant. So find the part on the diagram that says female. And then the pollen, beware, pollen is just like sperm. So if you've ever smelled a flower and gotten some pollen on your nose, haha, you got plant sperm on your nose. Okay, so this is what, oh, sorry. And the answer to that question was, if you cross the purple and the white flowers, 100% of the babies were purple. What? What happened to the white color? They weren't even lavender. They're not even a blending. They're straight on purple. And next to it, it says F1. That's called the F1 generation. So we had the P1 generation. Those were the original parents. We mated them, and now we have the F1 generation. And now we're going to look at the F2 generation, the generation that comes after them. But let's talk about how Mendel was crossing these plants. So what he did, usually it's bees and butterflies and other insects that carry the pollen, the plant's sperm, and they carry it from one flower to the next, and they fertilize the egg that's stored inside of the female part called the carpel of the flower. But to stop that, he instead took a paintbrush and he got the pollen on the end of the paintbrush, and then he went and painted on other flowers. So he carried the sperm, the pollen from one flower over to another flower, and that's how he was crossing them. So it says, then he crossed these purple offspring. So the F1 generation, he crossed them. So look at the diagram. There was a white parent, a purple parent, and when he crossed them, what did he get? All purple babies. So that's the F1 generation. So now the question is, what's the F2 generation? If we take two of these flower babies and we cross their pollen and their eggs, what kind of babies are we going to have then? And I want you to hypothesize. Will they be all purple again? Will they be all white this time? Maybe purple and white, maybe stripy, maybe lavender. 75% of these second generations. So we had the parents, we had the F1, and now this is the next generation. 75% of them were purple, 25% of them were white. All of their parents were purple. Where did the white ones come from? Why were they gone for a generation and then reappeared? Some of you might remember this from like 7th grade biology. That's awesome if it's a review, and if it's not, it's exciting to learn new stuff. All right, so to understand the results, we need to review a little bit of vocabulary. First piece of vocabulary, a gene. What is a gene? It's a part of a chromosome. Chromosomes are made of DNA. So y'all have heard of DNA. And when DNA twists up, it makes chromosomes. And this image, those are actually pictures of chromosomes. They've been purposely colored by a computer so that they're easier for our eyes to identify. So a gene is a part of one of these chromosomes that codes for one characteristic, one trait, or really it's a protein. Who cares? Protein sounds boring. So it just determines what your eye color is or whether your hair is straight or curly. That's what a gene is. Humans have 20 to 25,000 genes and 46 chromosomes. For each trait, we receive one gene from mom and one gene from biological mom and biological dad. Now, there's another word that I don't use very often, but the word is allele. And so an allele is an alternative version of the gene. So for example, if we have, I can't remember what it says here. Yeah. So we have the purple and the white color for the flowers. Those are controlled by different alleles. It's still a gene. The gene is for color, and the allele is purple or white. There's two alleles for that gene. It could either be purple or it could be white. All right. So, and let me back up. This photograph that we're looking at, let me get my picture out of the way. This photograph is really of somebody's chromosomes. And the chromosomes are kind of messed up here. So it's very possible that this is actually, it's called a karyotype. When you see, when scientists mix up all their chromosomes, they line them up in order. It's called a karyotype, and we can look at it and we can see some things about this person. And I will tell you that this probably came from a baby that died. And the reason I know that is because there's lots of flaws in the DNA. In particular, will you look, first of all, look at the first pair, chromosome pair number 1. There's two gold chromosomes. They look like letter X's. One of them's a little bent. That's okay. They have the consistency of like limp spaghetti noodles, so they bend, and it doesn't matter if they're crooked or something like that, but it matters if there's a part of them missing or if they're stuck together. That's what we're concerned with. So we look at chromosome pair number 1. One of them came from mom, one of them came from dad. Looks good. But now look at number 2. We've got a teal chromosome. Looks good. And then what's the other chromosome? It's like 4 chromosomes stuck to each other. Ooh, that's such bad birth defects in that case that's probably not a survivable condition. That would just be my guess. And we can look throughout and see some other problems with this individual's DNA. So tragic in this case. Let's look at another one. So now this one hasn't been colorized, but it's still called a karyotype. It's a photograph of somebody. It's a real photograph of somebody's DNA. So the question here is this person a male or a female? And do you notice anything surprising about this person's chromosomes? So as you look through, I want you to see that some of them are bent over. That's that limp spaghetti business that I'm telling you. It's okay that they're bent over. What matters is if there's the wrong number of them, or if they're attached, or if parts of them are missing. So I look at chromosome pair number 1. The first one came from mom, the second one came from dad, or vice versa. But there's 2 of them there. They look about the same length. I don't see much exciting. Pair number 2 looks good. Pair number 3. If we look all the way across, we see that there are 22 numbered pairs. And then the very last 2 are labeled X and Y. Who remembers what's X and Y and what that tells us? So it tells you whether it's a male or a female. So a male is XY and a female is XX. Now that's biological. That's nothing to do with gender, but because remember, gender is how a person feels about themselves, whereas the chromosomes are more about just biologically. What their genitalia will be, that kind of thing. Okay, so this individual was biologically a male. What I see that's a problem with their chromosomes, look down at the pair for number 21. You're supposed to have one from mom and one from dad. What do you notice about that one? There's 3 chromosomes. That's actually a birth defect. Some of you might be familiar with it. It's called Down syndrome. Anytime that anybody, an animal, a plant, and anything, anytime that they're born with the wrong number of chromosomes for their species, there's always a syndrome associated with it. It's always a birth defect or something goes wrong. And so Down syndrome is a fairly that's a funny way to say it. It's a survivable condition. Kids don't die when they have that extra chromosome. And that's because that extra chromosome is really, really, really tiny. But when kids have like an extra chromosome number 1, it's got so much information on it. It's our biggest chromosome. That's a fatal condition. So babies that end up with an extra chromosome number 1, they die. And there's not very many cases where somebody can have an extra chromosome and survive. So this child it's a male and he has Down syndrome. So he has 47 chromosomes instead of the normal 46 chromosomes that other people have. All right, moving on. Let's practice some more vocabulary. Genotype. Genotype is referring to the genes, and it's how we describe the genes using letters. And if you've ever done a Punnett square, you've heard of genotypes and you've seen them before. So the dominant gene, you may have heard of dominant and recessive. A dominant gene is a gene that can hide or mask another gene. So it's more important, it dominates and the other gene gets hidden. And we show that with a capital letter. So with the purple color, for example, purple, well, maybe I won't give that away because that comes up a little bit later. Let's choose something totally different. How about tall and short? Let's say tall pea plants are dominant to short pea plants, which is true. So tall is designated with a capital T. Now the recessive trait, a recessive gene is a gene that gets hidden by a dominant gene. The only time you ever see somebody with a recessive trait, or an animal or plant with a recessive trait, is if they don't ever have that dominant gene and they have 2 copies, one from mom and one from dad, of the recessive trait. And we show the recessive trait with a lowercase letter. So if capital T was for tall, lowercase t is for short. And the only way that you would have a short pea plant is if we had 2 lowercase t's, one from mom and one from dad. Okay, so take a look at the diagram or the picture here. It says, what is the dominant color trait for these flowers? Well, you can tell by what the capital letter is. The capital letter is R, so the dominant trait is red. Here's something I forgot to say. When we use a lowercase letter for the recessive, we use the same letter of the alphabet as the dominant. So here, white is the recessive trait, but notice we use a lowercase r for that. If you do sign language, that's the letter R, not that, but I was making a little R shape. Anyway, so it says, what's the dominant color trait for these flowers? And the answer is red. And then it says, why does the big R, little r flower look the same red color as the big R flower? Look at them. They both look red. There's a big R, little r, and there's a big R, big R. Why do they both look, why isn't the other one pink? They don't blend. The red, the capital R, hides the other trait. And so you don't see any white, you just see red. So we've got 2 red flowers, and the only way to see a white flower is if there's 2 lowercase r's. All right, phenotype is a physical description of the observed traits. So genotype is what we just learned. Now we're learning phenotype. And I say the hard to help you remember. It's the physical description of what it looks like. So in this case, phenotype is purple and white of these flowers. So it says, what are the genotypes and phenotypes for these flowers? So the phenotypes, purple, phenotype, white. We got that. That's easy. The genotypes are a little bit trickier. So purple is the dominant trait over white. So we're going to use a capital P. So some of the flowers could be capital P, capital P. Some of the purple flowers could be big P, little p. And then the white flowers have to be little p, little p. Remember, we're using the lowercase letter of the dominant trait. All right, and then this is the last set of vocab, and then we're set to go. Homozygous. Now, you may have learned that the prefix homo means same and hetero means different. I want you to keep that in your head because that's really going to help. So it says homozygous is when the genotype has two of the same letters. So take a look where my picture is. Where it says homozygous, there's two little r's or two big R's. That's homozygous. Does it have anything to do with homosexuality? Nothing at all. It just means that they got two of the same genes from mom and dad. Mom gave one recessive and dad gave the other recessive. It's homozygous. So you can guess heterozygous means that the offspring got one big R. I'm trying to figure out a big way to make a big R. I don't know. And a small R from the other parent. And that's heterozygous. So I might give you a question where I would say, what would happen if a homozygous recessive flower was crossed with a heterozygous flower? And then you would have to be able to answer that question. We're going to learn how to answer those questions soon. All right, so it says now back to Mendel's purple and white flowers. In the first generation, the P1 generation, we had purple and white flowers. 100% of the offspring were purple. Why? 100% were purple because the purple flowers were big P, and when you cross them with white flowers, little p, each one can hand off one gene. So the purple flowers donate a capital P, and the white flowers donate a little p. I'll make it little, a little p. And so there's always this dominant purple flower, this dominant purple color that's there, and it hides the lowercase white flower. And so we never see any white color. So 100% of their babies looked purple because they all had inherited one purple gene from the parents. Now, listen to that carefully. They had one purple, so they have one capital P, the babies, and they have one lowercase p. Now, let's try this again. He then took that all purple generation, which is called the F1 generation. They had a capital P and a lowercase p, and he crossed them with themselves. So a purple times a purple, big P, little p, crossed with a big P, little p. This time, only 75% were purple and 25% were white. That's tricky. Why is that? That's because the first each flower can donate one letter. They can donate a capital P or a lowercase p. So you could have some flowers that get a capital P from one parent and a capital P from the other. You could get some flowers that get a capital P from one parent and a lowercase p from the other. That's purple still. You can get some flowers that get a lowercase p from the first parent and a capital P from the second parent. That's still purple. And then you could even get some flowers that get two lowercase p's from the parents, and that's going to look white. So 75% of the second generation, they're going to look purple, and 25% of them, they're going to look white. We're going to learn how to do Punnett squares, and you'll see how to solve those kinds of problems. All right, I think that's it. That's it, everybody. Have a good rest of the night, and then I will make a Punnett square video so that you can watch and learn how to do the Punnett squares.